Hello, everyone, and welcome to today's webinar, Breaking Sample Type Barriers with Direct PCR. This webinar is part of the Cancer Research and Oncology Virtual Event Series 2024, brought to you by LabRoots, and is sponsored by Thermo Fisher Scientific. For more information on our sponsor, visit thermofisherscientific.com. I am Israel Gonzalez of LabRoots, and I'll be your moderator for this webinar today. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by both communicating with other attendees using the group chat and submitting questions during the presentation. Also, during today's presentation, we will stop to ask a few polling questions along the way. We would greatly appreciate your participation in answering the questions as they appear. Now let's get started. I'd like to introduce our speaker, Marija Navickaitė, Product Manager, Molecular Biology at Thermo Fisher Scientific. You can view Marija's complete biography below. Marija, you may now begin your presentation.
Warm welcome to today's webinar, Breaking Sample Type Barriers with Direct PCR. Here is an outline of today's webinar. I will present what Direct PCR is and its benefits, then how it works and what technological advancements enable Direct PCR. And the last part will be about troubleshooting and tips when working with this PCR method. So let's begin. What is Direct PCR? Conventional PCR workflow here on the left consists of multiple steps and is time-consuming. DNA must be purified, then PCR is set up and run. The whole workflow can take from 2 hours to half a day, depending on the methods used. On the right is Direct PCR workflow. So here, sample is directly added to the PCR master mix and amplified. In this case, time and resources needed for DNA purification is eliminated and hands-on time preparing for the PCR is minimized. So PCR workflow can take less than an hour, bringing you savings in time and resources as well. Now let's look who could benefit the most from Direct PCR approach. It is for those researchers who do endpoint PCR and analyze fragments on agarose gel. It can be any sample type and any tissue type coming from animals, human, or plants. Direct PCR is also a great time saver for high throughput labs since no purification is required. Overall, Direct PCR is ideal for yes or no analysis like genotyping, colony screening, and it can also be used in CRISPR workflows to confirm modifications. While Direct PCR workflow is fairly simple, there are challenges behind it. Reaction conditions are suboptimal for polymerase to work. Buffer helps lyse cells and release DNA from the samples. But cell lysis also releases proteins, lipids, and other cellular debris, which can inhibit PCR. Inhibitors coming from the sample can either affect the polymerase performance by altering enzyme structure or inhibiting uptake site, or even degrade the enzyme. And inhibitors can also affect DNA template, its structure, or prevent optimal primer annealing, or polymerase DNA complex formation. That is why polymerase tolerance to inhibitors and optimized buffer systems is critical to achieve robust and specific amplifications in such suboptimal conditions.
We would appreciate your participation by answering a few polling questions. To record your answers, select from the options on the screen, or in the case of this first question, type your answer in the text field below. Our first poll question is, name the model of thermocycler that you are currently using in the lab. We will give you a few moments to write out your answer. Thank you for your participation in this poll question. Marija?
Now, the Direct PCR approach usually offers two protocols to choose from based on experimental needs. The first direct protocol is very straightforward. A piece of the sample is directly added to the master mix, and you can start amplification. This protocol has minimal steps. It's quicker, but in this case, you can run one PCR from one sample. And here, sample size is very important because too much will inhibit the reaction. Second protocol is lysis protocol. It is more flexible, but has several additional steps. Here, sample is first added in the lysis buffer, and then supernatant is used for PCR. Sample size is not very important because lysis buffer volume can be adjusted accordingly, and this way you can run multiple reactions or repeat experiments. In addition, samples can be stored in the lysis buffer for some time. So overall, two protocols provide flexibility based on your experimental needs. Today, Direct PCR has come a long way. There were many technological advancements that, when put together, enabled Direct PCR. One is the polymerase resistance to inhibitors that enable direct amplification from a wide variety of samples. Universal primer annealing minimized optimization needed for different primer pairs, and I will talk a bit more about this later. Advanced buffer compositions enable amplification of challenging GC and AT-reach sequences. Hot start modifications now enable room temperature stability, which is especially important for high throughput research and reaction specificity overall. And of course, it is always important for any PCR experiment to have results of high yield, specificity, and sensitivity. In this webinar, we will be looking into Platinum Direct PCR Universal Master Mix capabilities and technological advancements that enable robust PCR. Now we can take a look where Platinum Direct PCR Master Mix was used to amplify DNA fragments directly from different tissues of multiple animal species. So the first one is different human tissue types, where this direct PCR master mix amplified from 350 base pairs up to 7.5 kb fragments. In the second image, you can see different mouse tissues, where again up to 3.6 kb fragments were directly amplified. Soft tissues were cut into about 1 millimeter size pieces, while hard tissues like bone has to be first crushed with a grinder to a small, about 1 millimeter size pieces. The next one, zebrafish. So here, successful amplification was achieved using fish fin, scale, muscle, as well as brain samples. Fruit fly was also tested. Now, direct amplification was done from even the whole body of the Drosophila, as well as head and larvae. Last but not least, FFPE samples. So here we managed to amplify up to 1 kb fragments from mouse kidney FFPE blocks. FFP DNA is often fragmented, and quite often we see that the sample quality is quite low. That doesn't allow you to amplify longer than 1 kb fragments. So one master mix enable you to achieve efficient amplification of different sample types. Here is just an example, a list of all the samples that were successfully tested with platinum direct PCR master mix. You can see here samples from mouse, human, tissues, plants, different bugs, and fish samples as well. So I've previously mentioned universal primer annealing. So let's see what it is in more detail. A new technology that has come in recent years, universal annealing temperature. This allows to run PCR at universal primer annealing of 60 degrees. As you can see in this gel image, each primer pair had a different annealing temperatures ranging from 48 to 68 degrees. However, using platinum direct PCR universal master mix, all targets were amplified using 60 degree as annealing temperature for all the primers. And you can see high specificity and yield of all different targets amplified from various samples. So this is a really game-changing technology, as you do not need to optimize primer annealing temperature anymore. And also this enables co-cycling of multiple targets simultaneously. When preparing for direct PCR experiments, other PCR capabilities should be considered as well. So first, let's look into sensitivity. This is an important feature if you are working with limited sample quantity. For example, here, 2 targets were amplified from 5 to 500 HeLa cells, showing how sensitive direct PCR is. So when choosing a kit for direct PCR, do check sensitivity levels that you don't have to worry about it when working with limited quantities. Long PCR can be a challenge as well. However, novel advanced polymerases enable you to run faster PCR along with amplification of long targets. In this example, 2 kilobase fragment was amplified using direct protocol and 8 kb fragment using lysis protocol from human blood sample. Amplifying GC or AT-reach sequences can be tricky. Thus, if you are working with such targets, look for master mixes that has capabilities to amplify such fragments effectively. And in this case, you can see an example in the image. 14 different targets with different GC content were amplified with high specificity with platinum direct PCR master mix. And one note that targets above 65% GC content may require an additional GC enhancer to achieve the best result. When working with high volume samples, especially where automation is involved, reaction setup can take a long time. And if reactions are left at room temperature, this can result in non-specific amplification and low product yields. Today, hot start technologies enable room temperature reaction setup without sacrificing end results. Hot start modification inhibit polymerase activity and stabilize the reactions at room temperature. Only when PCR starts, the polymerase is activated and can start amplification from the target sequence. In this example, reactions were left at room temperature for 8 hours, and you can see the result is no different from the reactions that were run right after the setup. Now this example shows suitability of the platinum direct PCR master mix for high throughput applications.
It is now time for our second and final poll question. To record your answer, select from the options on the screen. Are you planning to purchase a new thermal cycler in the next 6 to 12 months? A, yes, or B, no. Thank you so much for your participation. Marija, back to you.
And now I would like to show you an application example, how direct PCR can be utilized. In this study, platinum direct PCR master mix was used to identify bacterial genera directly from lake water samples. Direct PCR and PCR with extracted DNA from different samples gave similar results with 16S ribosomal RNA gene amplification, indicating high sensitivity and robustness of the direct PCR method. Then this was followed by next generation sequencing, and it was seen that read frequencies were more broadly distributed in directly amplified samples compared with extracted DNA, indicating that higher diversity can be identified with direct amplification. In addition, more bacterial genera were identified by direct amplification, suggesting that some sample material may have been lost during DNA extraction. So taken together with a direct PCR approach, can be used in metagenomic analysis of water samples, simplify the whole process, and even improve the chances of discovering a broader range of bacterial species. So lastly, I would like to give you some tips and tricks when working with direct PCR. So first, you need to be very careful about the sample size that you are adding to the reaction mix. It is commonly thought that if you add more of the sample, you can get better amplification or yield. However, that's not necessarily true when working with direct PCR. There are guidelines available, like in this image, what sample size or liquid sample volume can be used with different protocols. So always remember to double check to avoid failed experiments. So we can look on a few recommendations of sample sizes. For example, for human saliva, the recommendation is to use about 1 or 2 microliters of the sample, while for harder tissue samples like mouse ear, recommendation is from 0.5 to 1 millimeter size. In case of plant tissues like plant leaves, it's a similar recommendation up to 1 millimeter. When, for example, for organisms like fruit flies, half or even the whole body can be used for amplification. Now, post-amplification, gel electrophoresis is another step, which can be tedious and time-consuming, especially for high-throughput analysis. Now, our suggestion is to use advanced systems, like in this case, eGel PowerSnap electrophoresis system with precast agarose gels, which can allow sample loading, running, and documentation in as little as 15 minutes, depending on the throughput and the eGel used. So here, you do not need to prepare the gels and can run and document the results in one instrument. So last but not least, an overview of different applications where direct PCR can truly simplify and shorten the whole workflow for you. Applications like genotyping benefit from direct PCR because of the short protocol with no DNA purification. Colony screening after cloning or after CRISPR modifications. All these screening type of workflows are ideal for direct PCR, where DNA purification can be omitted and results are analyzed on agarose gel. So this is the end of today's mini webinar about direct PCR. We hope you enjoyed the time. Now we will open for question and answer session, and if your question is not addressed here, we will be sending you answers via emails.
Thank you, Marija, for that informative presentation. Although our speaker will not be available to answer questions live today, we will forward any questions from today's broadcast and from the on-demand period to our speaker to answer offline. We'd like to thank Marija again for her time and important research. And before we go, I'd like to say thank you to our audience for joining us today and to our sponsor, Thermo Fisher Scientific. This webcast can be viewed on demand for 1 year until October 2nd, 2025. LabRoots will alert you via email when it's available for replay, and we encourage you to share that email with your colleagues who may have missed today's event. Until next time, goodbye, everybody.